Hi, my name is Elias Georgas, and I'm a PhD student in Dr. Chin's lab. And the title of my talk is the mitigation of osteoclast formation and Netrin-1 secretion using low-intensity ultrasound. Knee osteoarthritis is a multifactorial degenerative disorder that affects 33.6% of the elderly population, which results in about $186 billion spent annually. And on the left here we have a healthy joint, and on the right here we have an OA joint. And what happens during OA is that there's cartilage degeneration, so this blue cartilage here starts to wear away. And then on the bone side, we see subchondral bone plate thickening, which is this region here, which results in ectopic bone formation. And then later in OA, there's dramatic bone loss. And then that also results in excessive angiogenesis and neurogenesis. And then also there's an increased inflammation in the synovium. And however, there is a diverse pain characteristic seen, whereas the stage of the disease doesn't actually correlate with the pain that the patients experience. And taken in all together, this disease has no disease-modifying treatment available. So that's why we're looking into low-intensity ultrasound as a treatment. An important part of OA is the role of osteoclasts and how they are involved in sensory innervation. So in OA, there's an increased expression of osteoclasts. There's more sensory neurons innervation into the joint. And then also in previous literature, a knockout of osteoclasts mitigated the pain behavior and sensory innervation. So during OA, the osteoclasts produce Netrin-1, which is shown in this yellow circle here. And the Netrin-1 is an important protein because it's an axonal guidance protein. And what we've seen in our previous in vivo data is that low-intensity ultrasound is able to mitigate the sensory innervation into the joint and also mitigate the osteoclast activity. So what my goal is to elucidate the low-intensity ultrasound mechanism and how it mitigates this osteoclast activity and sensory innervation. So my study was using the cell line called a RAW 264.7, which is a pre-osteoclast, which can be differentiated using the Rankel protein. And I have two groups here, which are as a control group with no ultrasound, and then a lipase group with ultrasound, with the parameters were 30 milliwatts per centimeter squared, 1 megahertz frequency, a 20% duty cycle, and the treatment was for 20 minutes a day for 7 days. And you can see this is the experimental setup here. So we have the cells in a plate in a water bath of degassed water, and the ultrasound transducer is placed 6 millimeters below the plate. And what we found is that low-intensity ultrasound is actually able to mitigate the formation of osteoclasts. So these big red cells are osteoclasts, and you can see there's a much greater difference there. And then what we see when we actually quantify, which is quantified by trap staining, which is this red, and a trap staining positive cell with more than 3 nuclei is considered a osteoclast. And we see ultrasound was able to mitigate the number of osteoclasts formed. So then we also want to look in the genes related to this. So using osteoclastogenic gene expression, we see that low-intensity ultrasound was able to mitigate almost every single gene except osteopontin, which osteopontin is a protein that anchors, the osteoclast is used to anchor to the bone, so it's not really as important in the actual activity of the osteoclast. And then also what we found is that we were able to mitigate the Netrin-1 secretion following the 7 days, and by day 5 there was a big difference seen. So this shows that we're able to mitigate the secretion of Netrin-1, which is the axonal guidance protein, and hopefully will reduce the sensory innervation into the joint. So taken all together, we found that low intensity inhibits osteoclast formation. So what this means, there's be decreased bone turnover, so that means there will be a mitigation of subchondral bone plate thickening. And then when we inhibit the osteoclast Netrin-1 secretion, with the decreased Netrin-1, there's a mitigation of sensory innervation. So my future directions will be to evaluate the role of Netrin-1 in neuron axonal growth, and actually evaluate the osteoclast neuron crosstalk following the low-intensity ultrasound treatment. And that's what I've been working on, and thank you for listening to my talk.